pretty proud. With that, I yield the podium to Sarah. Let me stop sharing.
Okay, thank you. Let's see if I can get this up. Window. Can you see my screen and hear me?
Yes.
Okay. Vasil, thank you so much for such a glowing introduction. President Tkach, ASP members, colleagues, and friends, I am incredibly humbled to be counted among the recipients of the Henry Baldwin Ward Medal. It is truly an honor to be here today, and I wish it could be in person. But I am thankful, at least, that we can use technology to gather, despite the devastation and uncertainty caused by our current pandemic. In preparation for today, I was looking through the addresses of previous Ward Medal awardees. In so doing, I found a common theme. Parasitology, it seems, is what happens when you're making other plans. In the tradition of many of these addresses, I'm going to share my plans, what actually happened, and a few of the lessons that I learned along the way. My path to biology actually began with art. In order to draw an organism and to make it identifiable, you have to understand its form and how to represent that form in a way that is unique from other similar forms. As an artist, you can use colors, shapes, and lines to create representations of each of these organisms you illustrate. Gross changes may easily separate distantly related organisms, but slight details may be needed to distinguish closely related species. Only recently have I realized that it is a small logical step to go from drawing distinct forms to wondering how those forms came to be. Evolutionary biology provides compelling answers to these questions, and in the final line of The Origin of Species, Darwin says, "From so simple a beginning, endless forms most beautiful and wonderful have been and are being evolved." Parasites are no exception to this. They are diverse, they are beautiful, and they provide exciting new opportunities to understand the evolution of biological forms. It took time, however, for me to discover parasites as the beautiful, evolving forms that they are. My parents nurtured an appreciation of nature. We went on many road trips and we camped a lot. As incentive for paying attention, we would actually build lists of species seen on each trip, and we would earn 10 cents for every new bird species on the list, and sometimes a quarter if we saw wildlife, like a moose or a beaver. My father taught us the Latin names of trees, although I later found out that many of the names he taught us had long been synonymized. Sorry, Dad. Even my favorite name, Pseudotsugo taxifolia, which I think rolls off the tongue, is no longer considered a valid name for Doug fir. Occasionally, if I didn't know the scientific name for something, I'd just make up a new one. I now know that this violates the International Code of Zoological Nomenclature, and I have learned to assign new names more properly. Our family would often head out early on a Sunday morning to the local woods or to the Red Rocks to paint. Watercolors in hand, we would spread out and paint for a few hours. During our mid-morning coffee break, we would line up all of the work, receive a critique, and then head out again to continue or start afresh. I preferred to draw organisms, flowers, leaves, birds, and bugs. And I especially liked getting close to capture the details. I was drawn to biology. In high school, I signed up for as many science classes as I could. To this audience, this probably seems like the logical progression. However, this was not the case. Why? Well, I grew up as a female in Utah. Even to this day, Utah routinely ranks last in the country for women's equity. Here is a Salt Lake Tribune headline from last year, and here's one from earlier this month. Women trail behind men in wages, leadership roles, and education, especially in STEM. And this is despite the fact that Utah has improved considerably over the last 40 years. There are many reasons for this inequity, but a large part of it stems from the patriarchal structure of the predominant religion in the area. I grew up in the county in Utah that is home to Brigham Young University, one of the main epicenters of the Church of the Latter-day Saints, or Mormons. Most of the community I lived in was Mormon. Most of the kids in school were Mormon, and even I was raised Mormon. Although as coffee drinking and sometimes Irish coffee drinking family, my nuclear family was certainly more on the liberal outskirts of this religion. When I was about 15, the bishop called me into his office. This was not a good sign. I was expecting a scolding for my mere monthly attendance at church or bad caffeine habits. Those topics did arise, but to my surprise, the bishop had my high school class schedule. He said, "I understand you're taking chemistry, biology, and calculus next year, but you're not signed up to take the LDS seminary course." Now, for those of you who didn't grow up in Utah, the LDS Church typically has a building adjacent to public high schools where students can attend an off-campus seminary class that just happens to be synchronized with the public school schedule. He looked down at me over his glasses and continued, "You do not need these science and math classes. You need to take seminary so that you can become a good little wifey." I was stunned. Wifey should not be a word, and it certainly was not something I ever wanted to become. I explained that I needed science and math more than I needed religion. I left his office and I left the church. Anger fueled my ambition. I was going to show the patriarchy how wrong they were. Women belong in science. I belonged in science. I was fortunate because I had parents, sisters, and teachers that supported my scientific ambitions. I was fortunate because I did not have to flee communist regimes like Vasil and Matt Bullock, both former Ward Medal recipients. It is important to recognize, however, that despite our free society, there are still visible and invisible barriers that prevent equality and prevent or deter people from pursuing their interests. We all need to encourage and support others around us. Even small actions can make a huge difference. For me, this came as a note from my high school teacher. Yes, this is my high school. We were the American Fork Cavemen. No apologies. My high school teacher, Mr. Reese, wrote on the top of one of my assignments one day, "You're good at this. Keep going." This simple note reached me at a time when I really needed it. It was confirmation from someone in science that I belonged. I have since had a chance to repay this note, and there is now a Chinese acanthocephalin named in his honor. I poured everything I had into studying in Mr. Reese's class. We covered the entire biology textbook, the Curtis and Barnes 5th edition, 3 times. We learned biology, we breathed biology, and we dreamt biology. In the entire class, my favorite section was about peppered moss, Bistonbetularia, and the change in color morph frequency that followed the Industrial Revolution in the United Kingdom. I was taken by the classic image of light and dark morphs attached to different colored tree trunks. This was a biological yin and yang, the light and dark forms oscillating based on conflicting forces of nature. The forms were beautiful, and the forms had function. Moss that did not match the background were more conspicuous, and they were more likely to be eaten by predators. The dark form was first reported in 1848. 47 years later, 98% of the moss in soot-covered Manchester were the dark form. The evolution of drastically different forms had happened within just a few decades. Of course, the story has gotten more complicated since I was in high school, but the story is still relatively simple, and I felt that biology had answers and that I wanted to help discover them. With encouragement from Mr. Reese, I applied for summer research programs. And thanks to the Howard Hughes high school program, I was placed in Rick Ward's lab in the Department of Human Genetics at the University of Utah. I learned cutting-edge technology then, yes, Sanger sequencing. And another high school student and I worked all summer to get DNA from human blood samples. I was ecstatic when I finally got a readable gel with more than 50 base pairs. And for those of you who can still read Sanger sequences, this is a picture of this gel that I had. And no, this was not the readable one. This was part of a serious research effort, and I was contributing sequences to a paper that ultimately included 360 mitochondrial DNA base pairs from 82 individuals from 3 different indigenous tribes. This seemed like so much data at the time. The project was using genetic data to trace the history of lineages and build phylogenetic trees. Other members of the lab were using similar methods to study the evolution of Bornean tree frogs or North American carnivores. I wanted to be a part of this research. Mistakenly, however, I thought that this research was called genetics, and it wasn't until much later, in a few wrong paths, that I discovered systematics, population genetics, and evolutionary biology differ dramatically from classical genetics. I applied to the University of Utah to go to school because in-state schools were what our family could afford. Luckily for me, the University of Utah was well known for its genetics programs. I had taken many AP classes in high school and started college with an honors at entrance scholarship and enough credit to start as a junior. I outranked the college student that helped me pick classes during registration, and not knowing any better, I followed his advice and took organic chemistry my first semester. That was not a wise move. I failed organic chemistry and I lost my scholarship. I did not appreciate the circumstance at the moment, but in hindsight, it was good for me. Scientists must learn to cope with failure, and this was a solid start. I began to focus more on the course content rather than on the grades, and I got a job in a lab so that I could pay tuition. I joined a genetics lab, led by a woman studying the transcription of bacteriophages. I did not understand what she was doing. Frankly, I still don't. She gave me piles of reprints to read, but I was lost. I spent hours, days, counting lac minus and lac plus blue colonies. I often wished that I was studying something bigger, something that had at least moved and had legs. Little did I know at the time that it would only be about 1 millimeter bigger and have 6 legs. My lack of fit was not lost on the PI, and after an unfortunate chemical spill in the lab, don't worry, it was just sodium chloride, she pronounced that I was too stupid to be any good at science. That was another blow. However, I had learned another lesson. Learning what you do not want to do is just as important as learning what you want to do. I reconsidered my options, and scientific illustration, I thought, might be a better career choice for me. I left her lab and started taking art classes along with my science classes. I started building an art portfolio. I met with the director of the local aviary and volunteered to design signs for their then aging exhibits. Although he didn't take my offer, he was very encouraging. He even suggested that I take the new ornithology class being taught by a new professor at the university. I went home and enrolled immediately. The course was great, and the professor, Dale Clayton, who studied birds and their ectoparasites, had space in his research lab for students. I joined, and ironically, the broader impact component of his new NSF career grant was to design signs for Tracy Aviary, and he wanted help. I was ecstatic. I ended up illustrating and researching interesting natural history tidbits, this is before the internet, for each of 100 bird species at Tracy Aviary, and I got paid. The Clayton Lab was very active in research, and I joined as many projects as I could, working with graduate students that had projects in the field censusing seasonal population dynamics of lice, or other projects involved with experimental manipulations using captive systems. I dropped my art classes and I redoubled my efforts in biology. One day, I asked Dale to teach me how to prepare bird specimens as museum skins. This is a technique used to create archival quality vouchers of avian specimens, which is important if you're trying to document host records. An odd look passed over his face, and he disappeared into his office to make some phone calls. 2 hours later, he emerged with a plan already in motion. Unbeknownst to me, he needed someone to help prepare birds during some upcoming field work. So he had arranged for me to spend spring break learning how to skin birds at the Field Museum in Chicago before heading into the field 1 month later for a 6-week collecting expedition in the Philippines. I agreed to this arrangement immediately. Specimens from the trip included 1 new species of bird and several new species of feather lice. I had no idea there were still so many new species to discover, and I had only just tipped the very edge of the iceberg. In the time that lapsed between checking nets and prepping specimens, or in this photo of Dale drying socks, it was a very wet field site. Dale and I talked about many different research ideas and potential graduate projects. By the time I got on the plane back home, I had an outline of a graduate project that was focused on studying competition between different species of ectoparasites. When I arrived at home, I submitted a late application to grad school and started an experiment to study competition between wing and body lice co-infesting rock pigeons. The experiment worked, and I found that different species of feather lice compete, and that this competition was mediated by host defense, much like Payne's classic work showing that predatory starfish mediate competition amongst mussels in the intertidal zone. As a graduate student, I also explored other factors governing host specificity, and I transferred 4 species of lice among 6 different North American dove species of different sizes and of different phylogenetic relatedness. I did crazy experiments to understand factors that might influence what could limit host switching. I attached lice to feathers on a high-speed fan to test whether or not lice might be able to remain attached to different size feathers during simulated long flights. A few of the lice got flung into my coffee, but on the whole, I found that lice were incredibly tenacious on all sorts of feather surfaces. Thinking that louse coloration may be a defense against parasites, I even collected louse frass to determine if lice were digesting or passing the pigments in different colored feathers. If someone had told me I was going to grow up and get paid to study louse shit, I would have assumed that that was an insult. Yet there I was, and I was happy. In the end, I found that a match between host and parasite size is a factor that would determine whether or not parasites could establish successfully on novel hosts. This relationship is governed in part by the ability of lice to escape from preening, when birds use their bill to remove debris and things like ectoparasites from their plumage. This is their bird's principal means of ectoparasite defense. Meanwhile, I had met, dated, and married in quick succession another student at the university. He was a political refugee from Iraq who had stood against Saddam Hussein and was trying to get back to medical school. Having left Iraq in the middle of his medical studies, I was impressed by his backbone, his stance against oppression, and his ambition. When he got a late season acceptance into med school in Grenada, he packed up and left for the Caribbean, and we began a long-distance relationship, each of us traveling back and forth when grad school and med school would allow. We kept it going for quite a while, but later during his residency, he tired of the circumstance and gave me an ultimatum: stay with him or do research. Our marriage ended shortly thereafter, but my graduate studies continued. As a graduate student, I learned a lot from being a teaching assistant in different courses: ecology, evolution, ornithology, and others. One year, I was asked to TA the plant systematics course. When asked about this odd assignment, as I had no real background at the time in plants or in systematics, the instructor told me that I was selected because I was the only student with any related experience, and that experience being that I had once taken botanical illustration. Throughout the course, I learned a lot about flowering plants and trees, phylogenetic trees especially. I learned that you don't really understand the material until you can explain it to others. And nothing focuses the mind like the panic of teaching material you have just learned to a horde of critical students. I learned that you only have to be a few lessons ahead of the students, but if you do not know the answer, do not try to hide it. Admit what you do not know. This is even more critical now that students can Google what you're saying during classes in real time. The more I teach, the more I am convinced that the I do not know lesson is incredibly important and may even be the most important lesson that we teach our students. As a society, we have trained people to memorize facts, and we have spent much less time on training people when to recognize the absence of data or how to recognize data that is not reliable. After I got my PhD, I became a postdoc at the University of Kansas Biodiversity Institute and Natural History Museum. I began working with Tom Peterson, who is leading an NSF biotic surveys and inventories project in China. It was a 5-year project, and each year, 2 ornithologists, 2 mammalogists, 2 herpetologists, and 2 parasitologists were to travel to the tropical region of southern China, a region considered a global biodiversity hotspot, to sample terrestrial vertebrates and their parasites. I was hired to coordinate the parasite collection effort. At this point, I felt really comfortable collecting ectoparasitic arthropods, but this is only a small part of what I was supposed to do. And I leaned on the members of ASP. Although I had been a member of ASP for some time, this is when I really felt truly initiated into the society. Scott Gardner invited me to the Mantor Lab, where Agustin Jimenez gave me a whirlwind training in the collection of helminths. It quickly became apparent, however, that a few days of training, even in the Mantor Lab, is no way to launch a top quality collection of helminths. We decided that someone with real experience needed to be on the expedition. And for some crazy reason, Don Duzinski agreed. A steady supply of beer might have been part of the negotiations, but I don't remember for sure. With a few phone calls and a lot of supplies, we headed into the field with our trusty companions. Here we are in the Beijing airport, getting our gear ready to head into the field. Fortunately, we had help getting it up into our field site where we had work tents prepared and ready to process specimens in really beautiful areas of tropical China. This really began my first full immersion experience in helminthology. A particular challenge of working in a group like this is that the ornithologists get up at dawn, they catch birds until dusk and prep late into the night, and the mammalogists and herpetologists do the opposite. The diversity of hosts that come through a base camp was amazing. It's like sampling from a fire hose. And it kept us parasitologists very busy. Don and I settled into a schedule with long hours, and Don patiently guided me through the dissection and preparation of hundreds of specimens from many different species. Over the 4 years of the project, we sampled over 2,000 host individuals of over 350 species. From these hosts, we collected ectoparasites, endoparasites, blood parasites of many different forms. This was, of course, too much for any one person to study, and back in the States, these specimens were quickly distributed to over a dozen institutions with specialists willing to work on these very different taxa. One particularly helpful person amongst this cast of characters was Vasil. I sent him some worms that I had collected in China, and he called out of the blue to let me know they could have been done better, he said. He gave me some great pointers, and he significantly simplified my field protocols, which I appreciated immensely. He even participated in some later trips himself. I fully admit that preparing helminths is an art form that I have not fully mastered, but Vasil is always around with encouragement and tips to do better, and I thoroughly appreciate his collaboration over the years. And as you've already seen this slide, I was able to name a species of louse after him in the genus Borrelia. On the personal front, I found that life is what happens when you are busy studying parasites. Dale Clayton and I, in Dale's words, decided to expand our collaboration. I'm delighted to say that this extended collaboration has worked quite successfully on both the personal and professional front. In 2008, Dale addressed this audience as he gave a speech accepting his Ward Medal. We are now the first married couple to have both received Ward Medals from ASP. Unfortunately, I missed Dale's Ward address because I was at home with the time with twins who were at the time of his speech just days old. I can, however, give an update. And as Dale predicted in 2008, we have indeed packed our kids off to remote field locations, and they have actually contributed to work in Utah, Slovakia, and the Bahamas, and soon will be co-authors on their first papers. Sonora is quite adept at removing birds from nests and even identifying lice, although she often proclaims that beetles, weevils in particular, are much more interesting than lice. Austin prefers the study of bird behavior and likes carrying around a spotting scope. It is not all work. We do manage to slip in a few extracurricular activities around periods of data collection. It's a different form of work-life balance than most people achieve. But it has been wonderful to work so closely with another biologist that understands the stressful moments, appreciates the importance of research, and can share the joy and the occasional misery of fieldwork. I joined the faculty at the University of Utah in 2010 and have continued collaborating with Dale Clayton on many new projects. Along with another collaborator, Kevin Johnson, we wrote a book summarizing decades of research in the bird louse system, and we put it in the perspective of other coevolutionary research. We anticipated that it would take a year, maybe two, to write. Fortunately, NSF didn't mind when our book came out 5 years later. We are all still talking to each other, which I count as a win, and we have several other ongoing projects. In addition to these two characters, I have other colleagues that I have not yet mentioned and can't possibly go through in detail. These collaborators and friends are incredibly important, and it's not just because of the expertise they bring to different projects. The roller coaster of a faculty position is similar to that of graduate school, but with higher highs and lower lows. There is more mundane paperwork, and other issues constantly loom on the sidelines, threatening to distract you from research. All of this is so much easier if you have others to encourage you through the grant and paper rejections, celebrate awards, or otherwise distract you with their exciting discoveries and new projects. I have been lucky enough to find kindred spirits in ASP and elsewhere. Thank you all for being encouraging, for providing letters of support, and for talking science over the years. Finally, as a faculty member, I have also learned that nothing can make research more fun and rewarding than a lab full of passionate people. I have been fortunate in having mentored such a group of students and postdocs. I have, of course, made mistakes in mentorship, and I thank each of these people for having weathered the process of working with me. I am proud of their accomplishments, and I am grateful for a chance to work with them. Here are the graduate students that I've had a pleasure to work with. Let me just give them a little bit of credit. Graham Goodman is one of our recent students from the lab. His work has been studying host parasite behavioral ecology, especially different behaviors that birds use to defend themselves against parasites like lice, such as scratching, which is a really underrepresented behavior in the study of parasite ecology. Amika Waite did a master's with me and studied the life history characteristics of Columbicola. Sabrina McNew now is a postdoctoral fellow at the Cornell Laboratory of Ornithology, continuing work that she started in the lab, looking at the effects that introduced flies have on Galapagos birds. These are parasitic nest flies that have devastating effects. Andrew Bartlow, who is now a research scientist at Los Alamos National Lab in New Mexico, has been studying parasite diversity in changing environments and is even a mammologist. He preferred to study rodents rather than birds, despite my efforts to convince him otherwise. Emily DeBlosity, who's now a research instructor in the Department of Psychiatry, actually is using her work, her training in population genetics to apply to human conditions now. She worked on population genetic structure of parasites on pigeons and on flies on pigeons around the world. Scott Villa is a graduate student who is later one of my postdocs. I'll talk a little bit more about his research later. And we now have two brand new students that have been in the lab for a year, Kyle Davis and Matthew Waller, that are still finding their way in different projects, but will probably be working on endosymbionts of lice and continuing work in the Galapagos. I can't tell you about all of their successes, but I'm going to highlight the stories of two people in the lab. The first is Daniel Gustafson. He was my first postdoc. He joined the lab in 2012 to work on the revision of a rather large genus of lice that no one had been crazy enough to tackle. In collaboration with Kevin Johnson and others, we took a genetically informed approach to species delineation. This made it much easier to identify phylogenetically informative characters, morphological characters that we could use to sort things more effectively. After many years of work, we published a rather large phylogeny to the group, and shortly thereafter, a monograph dividing the genus Borrelia into a complex consisting of over 20 genera and over 450 species. I knew at the get-go that we were a little crazy for tackling this project, but it wasn't until the completion of this monograph that I fully realized that Daniel is wonderfully insane. We've now worked on 3 major projects together, and we've co-authored 22 papers, with no indication that this collaboration is slowing down. Daniel is now a research professor in China, and our next project is just getting started. But I dearly miss walking into the lab in Utah and talking about lice, the natural history of any random organism, with him on a daily basis. Another project that I am particularly excited about concerns the experimental work of Scott Villa, who was a PhD student and later a postdoc in the lab. His work was a project focusing on the experimental adaptive radiation of parasites. One main aspect of his research, just one, mind you, was to transfer lice to different colored hosts, experimentally simulating a host switch. So you transferred the louse Columbicula columbae to different breeds of pigeons that varied dramatically in color. We were wondering if lice on these hosts would evolve cryptic coloration in the face of host defense. Birds preen their lice off as they defend themselves against these parasites, and they use vision during this process. So it's a perfect case to see evolution by natural selection. We found that lice did indeed rapidly adapt to these different colored novel hosts. And as little as 6 months, which is about 8 louse generations, we saw significant differences in louse color. And over the 4-year experiment, which is about 60 generations of lice, we saw dramatic changes in color that echoed variation existing across the entire genus. So let me just tell you what I'm showing you here. These are images of lice taken from the experiment. This image of louse in the middle is what lice look like on a typical kind of wild type feral pigeon. Lice transferred to lighter colored birds over the course of the experiment got significantly lighter. And indeed, here's the lightest louse found in our experiment. And lice transferred to darker birds were significantly darker. And here's the darkest louse recorded in our experiment. When you look at the variation across this genus, there are about 90 species in the genus Columbicula. They're found on many different species of pigeons and doves. Some of these species are very light, and some of these species are very dark. And the species of lice that are found on light pigeons are very light, and the species that are found on dark species of pigeons and doves are very dark. And what we found is that the color that we evolved in the course of this 4-year experiment pretty much echoed the color that existed in this genus that had been diverging for over 30 million years. In addition, we manipulated the ability of birds to preen by putting this C-shaped plastic bit between the upper and lower mandible of the birds. This is effectively a behavioral knockout and turns preening off. And when we did this, we found that lice transferred to light and to dark birds did not significantly change in color over the course of this 4-year experiment. Thus, in this experiment, we experimentally demonstrated that the rapid evolution of cryptic coloration by natural selection, and we demonstrated that host preening is the selective agent which preferentially removes lice that are conspicuously colored on the host plumage. What is especially satisfying to me is that this example now is included in many biology textbooks, along with the classic story of the peppered moss. We're not done with this project yet. Working with genomic collaborators, we now have a complete and annotated genome for the louse in the study, Collaborators columbae, and we are currently working to understand the genetic architecture of the phenotypic changes we observed among the lice in our experiment. Ultimately, the goal of this work is going to be to integrate across micro and macroevolutionary dimensions of evolution in order to understand how parasite forms have been and are being evolved. And with that, I would like to thank Vasil again for nominating me, and I would like to take time to thank the people who wrote letters on my behalf. And I would like to thank you all for taking the time to be here today. It's been a pleasure to be a part of ASP and to receive the Ward Medal. Thank you.